Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar, Behind the Scenes, making sure we don't have any technical difficulties. In this webinar, Dr. Kathleen Morgan-Kerr, Associate Director of QSP, will discuss how a license-ready QSP model of SLE can be used to explore different trial designs, patient subgroups, and a variety of dose schedule strategies. Samitra Ratakar, our Business Development Account Executive, will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all the questions in the Q&A after the presentation. Before we begin, I'd like to conduct a short poll to better understand your familiarity with QSP. And I'll go ahead and launch that now. And I'll give everybody a couple seconds to answer here. Okay, I see some answers coming in. Couple more seconds. And I'll end it there. Thanks everyone for your responses. And now I'll hand it over to our moderator, Sami, to introduce our speaker.
Hi, everyone. Thank you, Jasmine. It is my pleasure to introduce to all of you Dr. Kathleen Morgan-Kerr. Just give me 1 second. Jasmine, would you be okay if I share the results right now from the poll, or do you want me to wait?
You can share them. You can also close the window out when you're done.
Oh, sure. Yeah. So before I introduce Dr. Morgan-Kerr, maybe I'll just quickly summarize the poll results quickly. So about 7% of you said product differentiation, 27% said endpoint selection, 13% said locking down the clinical trial protocol, and all of the above was selected by 53% of the polling members. So definitely not surprising. We will be covering a lot of these points as we go along in this particular presentation. With that, yeah, it's a distinct pleasure to introduce Dr. Morgan-Kerr. A little bit about her. So Dr. Morgan-Kerr's work focuses on understanding the therapeutic modulation of the innate as well as adaptive immune response system in a variety of inflammatory diseases. These inflammatory diseases are challenging to understand due to the complexities associated with positive and negative feedback loops and other immunomodulatory mechanisms. This complex in silico work of creating the QSP model leverages known disease network, including both the immune and tissue cells, along with the known cell-specific responses that are used to simulate the clinical trial results. Prior to joining Simulations Plus, Dr. Morgan-Kerr worked at Immunetrics for about 14 years. And just so that you're aware, Immunetrics merged into Simulations Plus about the same time last year. At the legacy Immunetrics, she was involved with the development of several immunology QSP models for rheumatoid arthritis, psoriasis, psoriatic arthritis, ulcerative colitis, Crohn's disease, SLE, which is the topic du jour, including its affiliated diseases such as lupus, CLE, lupus nephritis, as well as early development of immuno-oncology models. With that, I want to request Kathleen to kindly take it away.
Thank you, Sami. Good morning or good afternoon, depending where you're at. As everybody's mentioned, my name is Kathleen Kerr. I'm the Associate Director of in the QSP Business Unit for Simulations Plus. And today I'll be talking about treating SLE and how QSP can support drug development and clinical trial design. Here's our disclaimer. Quite simply, SLE has an unmet need. Around the year 2000, a new class of therapies has entered clinical trials and subsequently the market called biologics. These are therapies derived from living organisms such as proteins, antibodies, or cells. Since then, there has been an explosion of new therapies for a variety of autoimmune and inflammatory diseases where antibodies directed against specific proteins are controlling inflammation. The success of these drugs is highlighted in the figures on the left, showing advances in arthritis and IBD approvals with various TNF inhibitors. This has resulted in a variety of treatment options for patients with these conditions. But when we move to SLE, it has lagged behind, with the first biologic, belimumab, only being approved in 2011. It took an additional 10 years for the second biologic, anifrolumab, to be improved with a number of other therapies that have failed in trial. SLP's tools in quantitative systems pharmacology modeling can be used to understand not only the disease, but also the patient populations. And when combined, these can be used to aid in designing clinical trials. Simulations Plus is a leading provider of modeling and simulation software and consulting services supporting drug discovery, development research, and regulatory submissions. In addition to QSP, we offer software and services in cheminformatics, PBPK, clinical pharmacology and pharmacometrics, as well as regulatory strategies. And most recently, we welcome proficiency into the family, which is involved in clinical trial operations. QSP modeling at Simulations Plus is a multidisciplinary effort. Our QSP group's technical staff includes 35 modelers. We have expertise in pathophysiologic, clinical, and pharmacologic aspects in treating numerous diseases, with a wide variety of training and backgrounds coming together to achieve multidisciplinary goals. There is a tremendous amount of collective QSP modeling experience in our group, including several original pioneers of the discipline. Simulations Plus has a growing library of existing QSP and QST models as listed on this slide. We are in the process of developing models for ovarian and endometrial cancer, as well as models for asthma and COPD. Today's webinar will focus on systemic lupus erythematosus, including a subpopulation referred to as CLE. QSP models predict efficacy via the intersection between pathophysiology, mechanisms, compound exposure, and PD. The foundation of our QSP models contain mechanistic representation of underlying biochemistry and immunology and altered pathophysiology. The next layer involves predicted compound concentrations at the target tissue using SLP, PK, and PBPK modeling software. Next, the models are informed by PD and mechanism of action effects and the interactions with underlying biology that is specific to each compound. Ultimately, SLP QSP models output the exact clinical endpoints used by regulators to examine therapies for approval in addition to biomarkers. Many of Simulations Plus's QSP models have been built using the Thales modeling platform, which was built to facilitate model development. A number of key features are outlined on this slide. A key differentiator of the Thales platform is robust support of virtual patient generation, as well as complex simulation pipelines, which allow us to replicate real-world clinical trial simulations. The combination of our approach and our tools results in clinically relevant modeling. Most often, Simulations Plus QSP models are used for prediction of trials involving a broad class of patients. We have found that fitting individual trials is insufficient for predicting response to novel therapeutics. Our models are simultaneously fitted to all relevant phase 2 and phase 3 clinical trials and their results to constrain a repertoire of our models. Our models are also built to include the necessary biology and clinically relevant details. Our outputs are not limited to biological readouts, but also extend to clinically relevant endpoints used for regulatory decision-making. This includes response criteria, as well as disease activity indices that are regularly included in our models. In addition to the inclusion of aggregate clinical data, our models are designed to represent protocols for relevant clinical trial design. This includes inclusion, exclusion criteria, use of concomitant medications, requirements for prior lines of therapy, as well as washout periods. This table highlights the number of trials, number of constraints, and clinical endpoints that are supported in our license-ready models. And when I say the number of constraints, this is simply the number of clinical trial data that is used to inform our model. This does not include any of the in vitro data and other training that the model is incorporated into our model. Digging into SLE, as any clinician will tell you, is a challenging disease from diagnosis through treatment. SLE is an autoimmune condition that involves up to 9 organs. And I say up to 9 organs because the number of organs can vary depending on which clinical scoring system is used to assess disease activity. This multiple organ involvement is a contributing factor to the slower treatment development that SLE has encountered versus other autoimmune conditions. Simulations Plus's SLE model includes the sites of primary activity utilized for evaluating tissue-specific disease activity in the skin and joints. This is because the first biologic, belimumab, when it was approved, post-hoc analysis identified that patients with higher mucoskeletal and mucocutaneous disease activity showed better response to belimumab. And more recently, we see the field is adopting this approach as we see skin-specific and joint-specific assessments like CLASI and swollen joint counts being reported as part of clinical trials in addition to general SLE endpoints. Simulations Plus's SLE model is highlighted, or the species that are included in SLP's model is highlighted on the right. And this model can readily be expanded as new biology is identified. The SLE field struggles with finding meaningful, achievable clinical endpoints. As a result, there are often new scores and endpoints being proposed. This table highlights scores that have been commonly vetted to be reproducible and used in clinical trials. Of the listed clinical endpoints in this table, joint counts like tender and swollen joint counts, as well as CLASI-based endpoints, are the most direct accessible for including in our model. Our model can also leverage proprietary individual patient data in order to better predict composite disease activity scores, and this allows the model to be extended and fit additional clinical trial data. The field is also slowly identifying organ-specific responses whose patients get their own trials, such as neurolupus as well as lupus nephritis. Clinical endpoints and assessments are only one part of the story. Another challenge in SLE clinical trials is variability. SLE trials are more complex than those for other autoimmune conditions. Some have required steroid tapering protocols, while others, those protocols are optional. They also vary in their mean and maximum steroid dose that is allowed. The consequence is shown in the graph on the right, where variability in the placebo SRI-4 response can be seen as having a 33-point spread across various trials. And this is a large challenge because the difference in treatment for these trials often was around 10%. Other considerations that occur in clinical trials include inclusion-exclusion criteria, can also be variable, with newer trials excluding patients with certain features. As I mentioned, neurological lupus and lupus nephritis are now getting their own trials. When we started developing the SLE model, we had a hypothesis that breaking down the placebos by trial design would help improve the detection differences between placebos and their treatments. To address this, our model was developed to have 6 different placebos. And we achieved these different placebos via this process outlined here, where the patient pipeline, virtual patients are generated and optimized while being simulated through the pipeline of scenarios. Patients start with a standard of care treatment, given 7.5 mg of steroids and antimalarials. Patients then undergo a steroid ramp up by increasing or decreasing their steroids until their symptoms have reached a steady state. At this point, patients are eligible for clinical trials, which then occurs in parallel. Patients are sorted into groups for the trials that they are eligible, and each group has its own placebo. This table highlights the drugs and trials that are available in the license-ready model. This outlines the scope of the data used to score the virtual patient generation and optimization. There are 1,261 mean constraints that include 45 trials. This includes standard of care reports that are not listed in the table, in addition to the drug arms. There are 29 arms, 87 analytes. In addition, there are model rules for heuristics on biomarker ranges for different types of patients, as well as flare distributions and frequencies. The model is further constrained by various patient parameter ranges, as well as identification of patient classification thresholds, like high interferon gene signature, as well as compartmental relationships and an SRI4 predictor. The specific use of this data between fitting and validation can be customized. We do have recommended approaches based on our experience, but each project is unique. This is an example of a model fit to one of our placebo arms, which does not include a steroid taper. As you can see, the model has good performance for C3, C4, and concentrations of anti-double-stranded DNA. Here is an example of a model fit that includes a steroid taper. And again, we're seeing good model performance capturing C3, C4, and the subsequent agreement with the reduction in class E50 and swollen joint count. Here is an example of a model fit to anifrolumab, which inhibits the type 1 interferon receptor. And again, we're seeing good model agreement and fitting to double-stranded DNA chained from baseline, as well as complement C3, C4, and the corresponding changes in class E and SJC. The SLE model has been built using in vitro data to inform lower level biology. And as I mentioned, it is fit to a variety of published clinical trial data. The model includes over 1,000 clinical data points in the license-ready model. And when we use proprietary individual patient data, the model can be fit to approximately 4,000 published data points in addition to the individual patient data. These additional data points allow us to fit to things like SLEDI as well as BICLA. And as I mentioned, the fitting strategy can be customized based on the project. We monitor the model fitting and validation using a combination of visual assessments and quantitative assessments to avoid individual modeler bias. Our quantitative assessments include calculation of the percentage of data points that fall within the model confidence intervals. The typical fit quality range for our license-ready model goes from 70 to 90% of the data points falling within the 90% confidence interval. And this depends specifically on the type of data being assessed. The graph on the right shows a model prediction for ianalumab, an anti-BAFF receptor. In this graph, the model was informed by the drug mechanism, but was not informed by clinical efficacy. And as you can see here, the model is doing a good job capturing these SRI4 responses for this clinical trial. Why is this all relevant? Here is a real-world example of the importance of choosing the right patient population and clinical endpoint. The story of litifilimab and daxdilimab. These drugs target plasmacytoid dendritic cells, or PDCs, which are a subset of dendritic cells that play a crucial role in SLE. These cells are responsible for type 1 interferon production, antigen presentation, B cell activation, as well as TLR7/9 activation. And there are a number of therapies that are emerging to target PDCs. At this point, this has resulted in a tale of two therapies: daxdilimab, which failed to meet its lupus study goals, and litifilimab, which was reported to be superior to placebo. To understand what happened, we start by comparing the two drugs and their clinical trials. They have slightly different drug targets, but both are expressed on PDCs. Both trials were phase 2, but they differed in their patient population, where daxdilimab recruited a general SLE population that excluded lupus nephritis as well as neurolupus, whereas litifilimab recruited CLE patients that could have had general SLE. They also differed in their clinical assessments. Daxdilimab was focused on patients who achieved BICLA response and oral glucocorticoid reduction at week 48. Litifilimab focused on SJC and Class A activity at week 16. These were obviously different endpoints and hard to compare. When we look at secondary endpoints, we found that both drugs recorded Class E50 in addition to other endpoints. So to compare apples to apples, we compared Class A and Class E50 between the two drugs. In this table, we highlight the different clinical efficacy between Class A at week 14 and week 16, as well as Class E50 at week 12 and 16 for both drugs. And what we find is when we look at daxdilimab versus litifilimab, daxdilimab actually had a greater reduction in Class A and a greater reduction in Class E50. This highlights the importance of choosing the right patients and the right endpoints. The importance of QSP in complex diseases. As we've discussed, SLE is a multifaceted disease that can manifest across many organ systems. It has multiple clinical endpoints, many patient populations, and many different trial designs, including entry criteria and steroid protocols. Issues for the trial designs are specific to the therapy: best steroid protocol, best max steroid dose, or should we target steroid tapering? SLP's clinically validated SLE QSP model can assist in designing the optimal trials for the specific therapy, exploring these factors. Any questions?
So before we get to the questions, I do have one more poll, and then I do see a couple in the Q&A. Let me launch the second poll. This one's just, are you interested in learning more about how we can help you with your QSP models? And I'll give everybody a couple seconds here. All right, I'll go ahead and end it there. And I'll pass things back to Sally.
Thank you, Jasmine. So very quickly, the poll says, are you interested, one more time, are you interested in learning about how we can help you with your QSP model? And yeah, some of you have answered favorably. And my request to you all is, would any attendee have any questions about the SLE model? Please type it away in the Zoom question and answer related window. Okay. We have a few questions. I'll give audience a couple of more seconds to put their questions in the window. Okay, one of the questions which we have from the audience is, how do you, can this particular model be used as a part of MBMA? Or can we tweak this particular model which Kathleen has described and create an MBMA model out of it?
I think MBMA and QSP are great tools that can work together. One MBMA requires understanding of efficacy from existing targets, and that is something that this QSP model can support. But the QSP model can also be built to explore drug targets that have not been tested in SLE, and that is one of the benefits of QSP, but they do work well together.
Thank you, Kathleen. There's one more question in the audience, from the audience, which talks about why do you think there are so many failures in the SLE clinical trials?
I see it as an evolving view of the disease. And I think a big part of the failure is trying to treat all the organ systems together. And because when you look at these scoring systems, they're weighted for certain efficacy. They're weighted for certain organs over others. And we also don't fully understand how long some of these organs take. Certain organs are faster at resolving inflammation than others. And I think that's part of what we're seeing in the current trial designs, where SJC, swollen joint count, and classy skin is also a very fast responding organ, as we can see in psoriasis. We also have a good handle on how fast to expect swollen joints count to resolve from psoriatic arthritis and rheumatoid arthritis. So those are two that we have a good handle on, and that's, I think, why the field is beginning to focus on these as clinical assessments. Things like neuro as well as lupus nephritis, understanding the dynamics of resolution of inflammation and how fast the organ can respond. That just, it muddies the water when you have a cutaneous lupus patient in with a lupus nephritis patient. And I think as we begin to tease that out, that's what we're seeing, is that, okay, we can lock down these organ systems. We understand how fast that resolution can occur, that inflammation can occur. And that will allow for focus on these other patient populations that are harder to treat, where the understanding of how fast the organ should respond as inflammation respond, we're just getting a better handle on that.
Thank you, Kathleen. There's one more question. How will you, I don't know how to phrase it, but this gentleman is asking about the digital twin topic and whether or not we address this particular digital twin.
So we, in theory, have digital twins. We don't have create patients that are mapped one-to-one to a real life patient. We have patient archetypes, but we can simulate our patients through multiple clinical trial pipelines. So that same patient can be simulated when eligible in multiple trial designs. They can show up there, so we can have that differential response. And our simulation platform allows us to go through and identify how does that patient respond in one scenario versus another. And we can also follow that patient through treatment failures and subsequent treatment efficacy. So we can treat them with another drug after they've already failed the initial drug.
All right, thank you. One more question. The gentleman is asking, can you speak to the relationship, correlation of the endpoints?
That has required a lot of research on our side. and is an extensive part of what we do and what differentiates us from our competitors. There's initially, when we discovered that SLE has moved to reporting SJC and CLASI, this was very similar to our experience with modeling rheumatoid arthritis and psoriasis, and was a natural jumping off point to extend the knowledge that we had developed from those conditions.
Got it. Thank you. One more question. How does the model handle, how does the model handle genetic information that might influence the disease? Could it handle novel information as the new research comes out?
That's part of why the Thales platform was built the way it was, so that we can readily extend the model. As far as genetic information, we typically would work with a client to figure out how the genetics modify the inflammatory condition. We have done this in other disease states with understanding of like HLA increases antigen presentation, or we have an increase in interferon alpha production, with a mutation for certain groups of patients. And so that's something that we can work with clients, understanding how does that mutation change the protein function. And sometimes we can also explore hypothesis testing if that function is unknown. Is that something that should be increased or decreased? Or it makes it challenging for drugs to target it.
Thank you. One more question. Does the model currently support clinical trial designs for lupus nephritis? What endpoints does it support, including, for example, GFR?
The model currently does not support lupus nephritis patients. That is something that we're looking into developing in the future. We plan to use much of the same inflammatory network that we have in our general lupus model, but it will require support of a kidney and the other related kidney endpoints as far as GFR, urine protein creatinine ratio, and to get a handle on renal response and remission. So those are things that we are actively looking into, but don't currently support.
Thank you. Next question, does the type of drug/antibody have any role on the model favorability? Example, chimeric versus humanized versus human antibodies versus inhibitors. Or is it purely based on clinical trial outcome data?
Right now, it's primarily based on clinical trial outcome data, but that is definitely a type of project that we can do with a client, which is if there is an understanding as to anti-drug antibody development and how that would affect efficacy, that is something that we can test within the model.
There's one more question on digital twin, which is, how do you foresee the evolution of digital twin methods over the next 5 years? Do you believe they will be more effective in certain therapeutic areas compared to others?
That's an interesting question. I'm unsure. I think it's going to be the evolution of the technology and what's available. I do think it will be an interesting thing to watch. Yeah, I'm not 100% sure how to answer that question. That's a good one.
Got it. And we see that you have developed, there's one more question. We see you have developed quite a few immunology models. So does Thales platform can help us in a pipeline in a pill approach, wherein we have one drug which we feel is effective across a variety of inflammatory conditions?
Yeah, that's definitely an interesting project to think about. We've taken that approach a couple times. We refer to it as like a pipeline prioritization to understand how one drug works, and we can test it in different disease models to see what the efficacy is, to help them prioritize where they will expect the most success.
There's one more question. Can you help in can your model help in clinical trial protocol design?
Absolutely. That's a big part of what this SLE model was built, and what we've found its greatest utility is helping understand the different clinical trial protocols, and not just the protocols, but the endpoints. There's just so many different approaches that the field is taking. And as I said, some of these things have muddied the water to understand truly what's going on. And QSP modeling is just a natural tool to bring all this data together and say, like, and force the model to reconcile how this all can function together. And that's actually the most exciting thing about this work, is being able to answer some of these questions that the field struggles with.
Thank you, Kathleen. There's one more question, which is, can you include, because I'll preface a little bit, there have been a lot of research for the new targets in SLE. So does the model support new targets as and when?
It's hard to say without knowing what the targets are, but the model can readily be extended to incorporate new targets. And that is something that we work on with clients to understand, what is it, a new cell type? Is it a receptor on an existing cell type that then we can use in vitro data that from preclinical experiments, and help them gain a quick assessment of how much they can expect, how much clinical efficacy they can expect from a clinical trial.
There are no more questions, Kathleen. Thank you for answering.
Thank you.
Thank you, Sammy. Thank you, Kathleen. If you do have any more questions that may come up, you can reach out to our team. They're going to provide answers for you after the webinar. We invite you to go to our website to learn more about our QSP capabilities. I put a couple links in the chat. You can also follow us on social media. If you or your colleagues may be interested in our upcoming webinars, we have one on ADMET Predictor, AI for pharmacological modeling, leveraging PBPK, PBM, and support of BCS Class 3 biowaivers. That concludes our webinar for today. The webinar has been recorded and will be available on our website and YouTube channel. Thank you again, everyone.
Thank you.